So today's lecture is quite short. It's just covering one last thing from the chapter 18. This is 18, right? Anyways, I forgot. Yeah, 18. And then that's pretty much it. So today we're going to talk about selfing, okay? So if you remember last lecture, we were talking about the assumptions that you had to follow with Hardy-Weinberg equilibrium. You need to remember that these must be in place, otherwise you don't consider the population as a Hardy-Weinberg, in Hardy-Weinberg equilibrium. That is being a large population, random mating. This is important, and it's going to be a differentiating factor for today's lecture, which is non-random mating. And then there's no significant selection, migration, or mutation happening on the population to affect this equilibrium. And then if we consider one gene with two alleles, then the frequency of the homozygous dominant is considered as p squared. Homozygous recessive is q squared, and then the frequency of the heterozygous offspring is 2pq, because it happens 2 times, right, in the Punnett square. So before we get into the questions, no, not the questions, into the selfing part of this topic, let's first just answer this one here. Anyone has an idea about this? No. So, yep. D? Nope. No? C. Okay, I put this here because I wanted to show a point here. Okay? Read carefully the questions, okay? Because a lot of wording like this, and then you start, I knew you were going to answer A and D, and I will tell you what's happening, okay? Let's start by A. Dominant alleles account for the most frequent phenotype in the population. When you read dominant allele in your head, you're thinking, yeah, probably it's the dominant one, so it's always going to be passed on to the offspring, and you will see it all the time in the offspring. But it does not work like this for a population that is in Hardy-Weinberg equilibrium, or even a population that is not, like humans. The reason for that is, even though the allele is dominant, the frequency that it is passed to the offspring is very small, and so the affected individual will not be the most frequent in the population. It will be the least frequent in the population. Okay, so this you can look at an example that I talked about in last lecture, and you'll see why this is not true. Okay, D. So the frequency of P for a dominant allele is equal to the square root of the frequency of the dominant phenotype. So who answered D? Nobody? You? Do you have a, do you know, what made you think that it's D? Yeah. Exactly. So you have to think here about not just one phenotype. You have to think about the heterozygous also, which has one of that allele, right? And so P is not really the square root of the frequency for the dominant phenotype, okay? So it's C. The frequency of Q for a recessive allele is equal to the square root of the frequency of the recessive phenotype. And if you go like this, and you look at this here, this is the rule I'm talking about. Yeah? Yeah. The dwarf example. Yeah. So, is it the same question? Something else? Okay. Let's remember that. Okay. So let's look at A first. So if you have a dominant allele, okay, so that allele overrules the other one, right? That allele should give you the most frequent phenotype, technically, right? Because it dominates the other one. The thing is, the most frequent phenotype is not always the dominant allele, because the dominant allele could be problematic. It could be, it could cause a disease, okay? Or it could cause lethality or something like this. So it's not the most frequent phenotype. The most frequent phenotype is the healthy one. And the healthy one could be either homozygous, right? Recessive or homozygous dominant. It depends on what you're talking about. And the other thing is, the most frequent phenotype is determined by the probability that one parent passing an allele that gives that phenotype times the other parent giving that allele, and then times the number of children they have. And so a dominant allele will end up getting diluted because of these probabilities from the two parents. Does that make sense? Kind of? Okay. Isabella, yes?
I don't understand why D is wrong, but C is right, because it's like the same wording, except one says Q and one says P.
Yep. So the recessive allele, so the frequency of the recessive allele is equal to the square root of the frequency of the recessive phenotype. So the recessive phenotype is what? Is two recessive alleles, right? So this is this guy here, right? Does that make sense? Frequency of these two together is this. So the square root of that will give you Q, which we learned is the frequency of the recessive allele, right? Does that make sense?
Okay,
and for exactly. So that's the thing here. Okay. No more questions. Okay. So what if we break the assumptions that we don't have a large population, or there's no selection, there are selection forces, or we deal with non-random mating? So we'll look at non-random mating. Non-random mating is just, well, it's clear. For example, there are two types of mating. You can look at two plants, let's say, for example, wheat or maize. Wheat is a self-pollinating species, so it almost completely always is inbred. So the plant itself pollinates itself, okay? So this is kind of self-fertilization. So we can consider this as completely non-random, okay? And it is homozygous for all genes because of this non-random mating. So if you remember, the more you mate with the same genotype, the more you dilute the heterozygous state and the more you produce the homozygous. And we'll see this in a second. Maize is a cross-pollinating species, and because it is cross-pollinating, it ends up having a higher degree of heterozygosity, okay? So this is a concept that we're going to cover today. It's called inbreeding. So this breaks the assumption that we have random mating for the, for Hardy-Weinberg equilibrium. And so a decrease in heterozygosity and increase in the fraction of homozygote individuals is different from Hardy-Weinberg, okay? So let's take a look at why this happens. So if you are dealing with a species that is self-pollinating, for example, this is an extreme case of inbreeding because you're basically breeding with yourself. And so with each generation, you will be losing heterozygosity, okay? And then the population will be out of equilibrium. And so if you take a look at this, if this one breeds with itself, it's only going to produce this homozygous dominant, right? And so with time, wait, I'll get to the frequencies in a second. The heterozygote here in this case will start breeding with itself as well. And because it breeds with itself as well, it will produce the 1 to 2 to 1 ratio. And so it will decrease the heterozygous state by 2. So that's why you see it starts with 2 PQ, which we learned right here, right? And then with one generation, it becomes PQ, and then PQ over 2, and then PQ over 2 to the power of n. And at the end, you'll end up with basically nothing here. Let's say, for example, you have 10 generations, 2 to the 10 is a huge number. And so PQ over 2 to the 10 is pretty much 0, okay? Because of this, the others, the two other states, will go from P squared and Q squared to about P and Q. And it's just from this here, okay? This, you don't need to go in depth about this. It's just to illustrate how these numbers change with each generation. Yep. Yeah. This one here? Yeah, so it's self-pollinates, so it will basically it's a cross between two dihybrids, two monohybrids. Okay. No, that's where. So when you do the Punnett square, okay, you're starting with two parents that are heterozygous, right? But then you will produce, let's say, 4 offsprings. 2 of them will be homozygous. So you're creating those homozygous and adding them to the population. Exact. And you're decreasing the heterozygous because you're adding those homozygous. You understand? Yeah. Exact. And at some point, you'll end up with very little heterozygotes. Cool. So this is a way that, people that are working with crops and all that, they use this to produce pure strains. Actually, we do even in the lab, okay? When we were working with mice and all that. But not this way though. You're not really self fertilizing there. You're using, similar backgrounds that are genotypic backgrounds. So if we look at the numbers, let's just take an example here so that you can see how this works in terms of numbers. So if we have a population with these genotypic frequencies, for the homozygous dominant 0.36, heterozygous 0.48, and homozygous recessive 0.16. So if we look at 2 series of self-pollination, you can see that when you produce the Punnett square for this, you will be reducing this by half of it, right? Because you'll be producing 1/4 of this genotype and 1/4 of this genotype. So you'll be increasing the frequency of this genotype and the frequency of this genotype and decreasing the heterozygous state. Does that look like something simple? Kind of? Okay. So then when you do that a second time, you see that you go from 0.48% to 48% to 12%. Okay. And if you do it one more time, you'll have nothing left. Technically, okay? This is all theoretical. Obviously, in populations, it's a lot more complex than this, but this is the basis of population genetics. So this is what I just said. If you realize, if you go to the store and you look at the vegetables that you're buying, they all look the same, and they're very uniform, and this is because of this. What they do is they just self them with themselves, and this creates kind of uniform crops because it will bring them all to this homozygosity, very hard word to say, and the plants will be breeding true, okay? And so they'll all have the same size, color, texture, and be sold together. So selfing is the way that you get to this point, and this is just another example. So we said that when you take a population, let's say a monohybrid, and you cross it with itself, then let's imagine like the whole genome, okay? So if you take 2, let's say, interesting tomato plants or wheat plants or whatever plant that you want, there will be homozygous for certain genes and heterozygous for other genes, right? And so if you cross them, you'll produce dihybrids for certain genes, right? That are different between the 2 parents. And so the first generation of those heterozygous will lose 50% of the heterozygous when you self them to themselves, okay? And so every subsequent selfing generation, the heterozygous state will become smaller and smaller and smaller, and you can see by round 10, you'll end up with almost no heterozygous population, okay? And so 99.9% of their loci will be homozygous. Cool. Just a visual way of doing it, okay? If you take plants, you self them. Let's say you take this one and you self it. And then after 4 generations, you take 1 sample plants, you'll see that it will have changed to the homozygous state for certain loci, for example, the B and the E. And then you sample it later on, you'll see that the others also change to a homozygous state. Okay, yeah. In the plants, yeah. Because you can't really self for humans there. But There are some animals actually that do that, some worms and all that. Yeah. Yeah, yeah, exactly. Because you'll be making like either the homozygous dominant or homozygous recessive and decreasing the heterozygous. Yeah. Actually, there's something similar to this for humans, and this is when there's inbreeding within the family, and we'll be looking at how that increases the factor of diseases. So look at that, it's the slide right there. So actually, inbreeding in humans will increase, and animals will increase the frequency of genetic diseases, as I said last class, because it will increase the size of the population. That's the first thing. It will increase the frequency of individuals that are homozygous recessive. And if you remember correctly, most diseases are caused by recessive alleles in the homozygous state. And it also increases the homozygosity for all alleles in the population. Okay, and this is a big problem, and we'll see how to calculate that. Okay, so there's something called the inbreeding coefficient, which is defined as the probability that two alleles are inherited from the parent are identical by descent. So what does this mean? So if you look at this family right here, really messed up family, but that's okay. you have this. So you have these two fathers and this mother here that basically get children together, and so you end up having half brothers and sisters. Okay. And so the probability that two alleles inherited from the parents are identical by descent, that means that the alleles inherited by those parents are identical by descent. That means they both come from the same parent. Okay? Just means that. Without any crossover, without anything. The DNA sequence comes directly from this parent to this one here, the same way it is in the parent. Okay? So when you have this offspring made from these two half-brother, half-sisters, this increases the risk of this disease by a lot because you have more chances of inheriting this bad allele, okay? And in this diagram, this parent is passing the red allele, let's say, which is the problematic one. And so when it is passed to this offspring here and this offspring here, and then they mate together, there's more chance of that having that disease passed to this kid. Okay, so how do we calculate this? Does that make sense what I'm saying? Easy? Okay. So F, the inbreeding coefficient, is, in other words, if you put it in a simpler way, it's the probability that I, this individual here, will inherit the same allele from both parents. So that's the probability that B will inherit the same allele from A as C. So this parent has half, like 50% chance of giving this allele, right? So half of the time that will go here. So that's 1/2. And then we have another probability that we need to take into account, the same thing, but for the other parent. So this allele has 50% chance of being transferred to the other parent. And then the chance that these two will give, B and C will give the same allele is 1/4, because 1/2 times 1/2, okay? And so if you want to calculate the inbreeding coefficient, you will multiply these probabilities. You always start, yep.
Is it like 50% that it gives to B and 50% to A, or 50%?
Both. So basically 1/2 here, 1/2 here, that's 1/4, right? And wait, oh no, sorry, we're calculating for, what am I doing here? 1 second. Let me think first, and then I'll explain. It's from, it should be 1/16, I think. Wait 1 second. So 1/2 here, and 1/2 here. Okay, so that's 1/4. And then B and C give the same allele. Okay, I think there's a mistake here. Okay, let's do something. Sorry, guys. It's not good teaching, but it's okay. Let's do it this way. So forget about that slide, okay? There's a mistake in it. Let's do it from here, okay? So let's restart from the beginning, okay? So the F, the inbreeding coefficient, is calculated this way. You start from the grandparent, and then you look at how a bad allele is transferred to the parent, and then from there to the offspring. So if you calculate the F, the inbreeding coefficient for the individual I, then what you're going to do is you're going to take the probability that this allele is passed to B and to C. So that's 1/2 times 1/2, that's 1/4, right? And then the chance that this offspring gets the allele from the two parents is 1/2 again and 1/2 again. So you multiply all that, because all this needs to happen for you to reach this offspring here. So that's basically 1/16. Yeah. Wait, can you repeat that, please? Forget about the first slide. There's a mistake. Wait. But B inherits the same allele as C. Okay, let me clarify this on my free time, and then I'll get back to you, okay? I'll send you an email today.
I think the other side was only talking about the maybe that's why I got confused.
Yeah, that's what I'm trying to understand, but that's...
Like the 1/8 is for B only.
Okay. Yeah, I see. Okay. So you're calculating here for one allele, and then here you're adding for two alleles. No. Yeah, first one is about just passing the red one in to the individual. Let me just see something here. Accidents. Okay, let me redo that. But you guys are right. Okay, so this slide, what it was talking about is just it's talking about just one allele being passed to the offspring, okay, from grandparent A. And then this one here is referring to the two alleles being passed to this offspring here, okay? And then at the bottom here, the calculation that that is happening here is just considering the other genotype for it to be homozygous as well. So the probability that I has two blue alleles is pretty much the same as the pink one or red or whatever color. And so you will just add them together, okay? And so this inbreeding coefficient, there are numbers that are already known for different combinations in terms of inbreeding, for example, half brothers and sisters or cousins and all that, and we'll see that in 1 second. But the meaning of F really, it's just the probability that one gene is identical homozygous by descent, so basically coming from here and becoming homozygous here. So that means that when we're looking at a full genome, F represents the fraction of genes that are homozygous by descent, okay? For half siblings, this ratio here is 1/8. So if they mate together, 1/8 of their genome will be homozygous, and you can see how this is a big problem in terms of diseases, okay? So let's take a look at another example where you're looking at full siblings, crazier stuff. So in this case, the ratio is even bigger, and there's more chance of getting the disease because you're increasing the chance of getting into the homozygous state. So if we look at this example here, and we have these two parents or grandparents that have a son and a daughter. So let's look, for example, let's say that A1 is the bad allele. So the probability that the offspring will be homozygous for A1 is 1/2 because, not 1/2, 1/16, because it's 1/2 to pass it to this son and 1/2 to pass it to this daughter. And then same theory here, the son has 1/2 chance of passing it to the offspring and 1/2 chance for the daughter. Okay, so when you calculate that, you will get 1/16, and then the probability of being homozygous for any of the 4 alleles, well, you're just adding this ratio here. So 1/16 plus 1/16, right? So you can see that it becomes 4/16, which is 1/4, and this is super big in terms of disease. Okay. So, right. Yep, here. So the inbreeding coefficients are the following. Self-fertilization, as we've seen, is the one that has the most chance of producing homozygous individuals, because every time you're decreasing the heterozygous state by 1/2, in this case by 1/4, etc. And cousins is 1/16. So just information for you guys, in Canada, it's actually, there are ratios like this the government uses to determine if it is legal or illegal. And I think the first legal part is the cousin, and then there you can go to second cousins, etc., and mixes of this. Okay, so the government does this because they want to prevent diseases spreading in the country, especially genetic diseases, because they're the hardest to treat. Okay, you can't really go and modify the genome. If you have a problem with, for example, diabetes type 2, you can do exercise and all that, but genetic disease is a bigger problem. It comes directly from the gene. Yep. Parent with the child. Some illegal shit. This happens. You can read a lot on this stuff here. Yeah. Sorry? The what? ODIP? Yeah, it's messed up. It's it's messed up. So these things happen. Obviously, not that much, but it's criminal. You can get like 10 years, I think, or something like this, if you are found to do that. But it is interesting in terms of genetics, because we use it for plants to create better crops and to direct genetics in terms of animals as well, right? It's better to use full siblings in terms of mice work, because it increases your chance of getting the homozygous that you want, compared to using cousins, right? So there's things like this that we do in the lab, okay? The mice there, they don't care. Okay, so what's the probability of an offspring being affected by a genetic disease as a result of mating between, well, inbreeding basically? And so we've seen the Hardy-Weinberg equilibrium formula. Now we'll try to apply it to this concept here of inbreeding, because we can also use those numbers in terms of inbreeding, okay? So imagine you have a population in equilibrium, and then stuff like this starts happening, and so you end up affecting the frequencies, right? So you can calculate these frequencies through the following way, okay? And this is an example for full sibling mating, so what we just saw. So usually you start with the grandparent first, because that's the one that's passing the allele in question, okay? And then you have the offsprings of that grandparent, which will end up mating and producing this offspring, okay? So you look always at the grandparent. And so the probability that that grandparent or the other is a carrier of that allele is 2pq plus 2pq, okay? You've seen this before. And then given that one of the parents is carrier, so the probability that that allele is passed to the son, it's 1/2. The other is 1/2, right? And the probability that the child is homozygous is 1/4, because you multiply this by this. You multiply this by this to figure this one out, okay? So in terms of numbers, we said that the probability that either, sorry, this is grandparent here, 2pq plus 2pq, and then if we calculate the whole probability of that offspring to get this disease, well, you'll just multiply everything. So 2pq plus 2pq times 1 over 2. So this is this 1 over 2 here, times 1 over 2, this one here, times 1 over 4. And then if you simplify this, it will give you pq over 4, right? And so, in other words, if you're looking at the probability of a child being homozygous for a diseased allele in a full sibling mating, it's pq over 4. Okay? A lot of numbers, huh? But what else? So we can use this to actually, so the way we're doing this right now is very simple, okay? It's actually a lot more complex than this, and this is just a basic example to show this, but technically you have 3 different cases that you have to consider. The first one is only one grandparent being the carrier and not the other. The other one is that the other grandparent is carrier but not the second one, and then both being carriers. And so when you're doing these calculations, in real life, you're taking into account all this stuff, not just the simple way that we've shown it here. Yeah. Yeah, but when you're calculating the total probability, you need to take both example. Yeah, exactly. And this is actually used. It's not just some theory that you're learning right now. If you go to genetic counseling and all that, they will study your whole genetics and try to do these numbers here to figure out the chance of you getting disease offspring or whatever. Yep. Sorry, this is just a simplified example of actually how to calculate the probability. Okay, because so here I'm just using an example for this thing here. I'm just separating the alleles like A1, A2, A3, A4 so that we can just focus on this one being passed. But you can also calculate the probability that all these will become homozygous, right? And you will have to count that through this and add them all together. Okay, but this is a simplified example. In reality, you take all the scenarios that are possible. You can also use a different way to calculate this by using the F factor, so the inbreeding coefficient. And so it's the same way as doing 2pq plus 2pq times the probability of homozygous by descent, which we learned earlier right here, right? So right here, times the probability that the homozygous by descent is the recessive one, so that's 1/4. And the F, we just learned it, it's the same thing as, so this here, P homozygous by descent, is the same thing as half times half, okay? Make sense? And then 1/4 will have the disease. So you can use both ways. Let's take a look at the, let's take a look at the, yeah. No, I will give you all these functions in the, you'll have a formula sheet for the final. Yeah, I can do that. I can send it to you. That's a good point. Okay, so let's take a look at cystic fibrosis in this design here, okay? So last class we were looking at random mating, and we saw that the rate of cystic fibrosis was 1 over 2,500. So the chance of you getting this in a random mating setting is 1 over 2,500. Let's take a look at a full sibling mating situation, okay? So again, we use this formula, which I just showed to you, to figure out the probability that the offspring will get this disease, right? And so we have 2pq plus 2pq times 1/2, 1/2, 1/4, which gives you basically pq over 4. So let's consider it from now on just pq over 4. So for cystic fibrosis, pq over 4 is 1 over 50. If you remember, that was the q last lecture. P is 49/50, but it's so close to 1, you just consider it as 1, okay? You don't break your head with this. Times 1/4 here, and that gives you 1/200. So you see how the risk of getting cystic fibrosis increases by 12.5-fold in a full sibling mating scenario. This is a lot. There are other diseases that are increased by a much higher number, okay? What is this one here? Child. So let's look at cousins. It's basically the same thinking. So for cousins, you have to follow this thing here. So instead of going through the whole 1/2, 1/2, 1/2, I'll just use the coefficient that we learned earlier. So cousins is 1/16, okay? So start with the same thing. So first, the probability that either the grandparents have it does not change, times the coefficient. The coefficient takes care of this part here, this part right there. And then times 1/4 takes care of this part here. 1/2, 1/2, and the coefficient takes care of these two passing here, okay? So that gives you PQ/16, okay? So I'll just remember PQ/16. Actually, don't even remember it, okay? You'll have it. So for cystic fibrosis, the rate of disease in the offspring for cousins mating is 1/800. So that's basically a 3.13-fold increase in the disease. Make sense? Good. Where are we? I thought it was going to be fast, but apparently not. Okay. So again, I'm reminding you this stuff here because it's very important in terms of Hardy-Weinberg equilibrium. Now let's look at other stuff that actually take off the population from Hardy-Weinberg equilibrium. Genetic mutations. Last lecture, we went like super quickly on the mutations, but I'll explain it a bit more in details now. So mutations affect this equilibrium because they change the pool of alleles in the population, okay? And the mutation rate is different for each species. You can see which ones mutate faster or not. SNP mutations just means single nucleotide polymorphism. In other words, a change in a single nucleotide, okay? Microsatellites, don't worry about it. It's just a repeat that gets deleted in the sequence, but not necessary to care about this. And so you can see that different organisms rate of mutation decreases, right? So in humans, we have a higher or lower rate than yeast, than E. coli. Higher? Why? So, yes. So there's more mutations per base pair, let's say, okay? There's more mutations that happen per base pair. That's pretty much what this means. So if you multiply it by 1 billion base pairs, you will see that you'll have, for example, in this case here, you'll have 4 mutations, okay? In this case, you'll have 30. Capiche? I think my math is good. Yeah. Yeah, pretty much. Humans and maize. What else? What else? But this is true, but not true at the same time, because humans, their cells replicate less often than bacteria, right? So bacteria mutate more because they have a faster reproduction cycle, and so they gain more mutations, right? It's just they happen less often within a certain time point. But if you let them replicate, they will generate more mutations than humans. Yeah, exactly. Statically speaking, humans are the ones that get the most. Yeah. Yeah, exactly. Yeah. This is after the repair mechanism. So when you get offsprings, your chance, so there's about 100 single nucleotide polymorphisms that are introduced in the genome, right? And so you would be like, okay, well, if there's only 100 in 20,000 genes, excluding the non-coding part. How come we are all so different? Don't forget, we have recombination and other stuff, okay? So this is one way to create variation in the population, but it's not the only way, okay? These mutations sometimes can be good, sometimes can be problematic, as you already know. What else? So when you have a finite population, the number of alleles will change in the population, okay? What do I mean by stochastically? It just means that it happens randomly, unexpectedly. Like you don't know. A stochastic process is like the stock market. You can apply statistics to it, make your lines there and try to invest at the right time, but you can never really predict it to 100%, okay? So this is what stochastic means, right? And so the stochastic process of increasing or decreasing the allele frequency in the population is called genetic drift, right? And so this also will affect the Hardy-Weinberg equilibrium, because the more the increase or decrease of these alleles will change that equilibrium, okay? And this is caused by genetic drift, so people leaving and coming in the population, right? The bigger the size of the population, though, the more linear it becomes. And the process becomes more linear, and so that the drift has less effect on a larger population, which is usually in Hardy-Weinberg equilibrium. If it's a smaller population, it is not in Hardy-Weinberg equilibrium, okay? Because one small change in that population will affect those numbers by a lot. Make sense? There's also something else that happens by chance. Well, not really by chance, but not really well understood, is that some mutations actually reach a frequency of 100% in the population. And this happens through many different mechanisms that together we just call chance, because we don't know how they come together, okay? But this happens for mutations that have no problem that cause no problem to you, that are deleterious, or also because of genetic drift, okay? So let's say, for example, for a reason, there's all the sickle cell anemic people move to Montreal, right? Well, and then the others leave, right? So it seems like chance, but there's a reason behind this migration, right? So that will lead to those mutations being fixed at 100%. Yeah. Yeah. So take, for example, birds. Sometimes they fly to different places, right? And so the first time they fly there, some of them will stay there and they will build this kind of population right there, right? Others will go back, right? And so with time, the selective pressures that are applied to both populations will affect them differently, and they will end up developing differently. And now these guys decide to come back. See these guys, well, now there's a change in the alleles in that sense, okay? Because both were subjected to different pressures and then came back together. Does that? Yeah, yeah, that's pretty much it. Yeah, exactly. So then you'll be passing that allele, right? And yeah, I will. I don't know why I give these long explanations, but yeah, that makes sense. Okay, fitness. We talked about fitness last lecture. So we were talking about in fitness. So there's selection for genes that allow you to live longer, reproduce, and then better chance of survival. And so these mutations will be selected for within a population. Everyone knows Darwin, so I don't need to introduce. But in population genetics, we use the term relative fitness. So this is just comparing two different individuals together, or two genotypes together, to see which one is better fit, okay? There's a way to calculate this. I'm just introducing the concept. We're not gonna do the calculation here, but it basically means how many times more offsprings of that genotype will be present in the next generation. And so the more it's present, the more fit it is when you're comparing alleles, okay? So there are mutations that can be advantageous and those will usually end up being fixed while the others will be removed. Okay, and this will increase these ones, the advantageous ones, have a higher relative fitness compared to the other ones, right?